everybody joining into this webinar. And Fabian will help me to guide me through the questions. I will focus on the talk. I would like to thank also all the members of DeCOI actually, because it has been quite a journey since March. We have a lot of people in Germany that work on omics have connected. And it has been work, but it has also been very enjoyable to see that there is many people in the country that really want to bring their knowledge and expertise forward to also contribute to a better understanding of this crisis we're all going through. This is then basically ended up in this initiative, and we're trying on several levels to basically contribute knowledge. And I will share you some of that today, which is more towards the single cell omics field. This is you see that it's really a lot. There's 60 groups from 42 institutions that are in there. It's really across Germany now. As I said, there's a lot of people in there, and what I would like to do is basically present you some of the work on the single cell level that we have. So the major goals is not only single cell. We have people that work on viral genomes and metagenomes. There's a huge group working on using omics technologies to understand the immune response. And then another very big group looking on the host genetics. And I think the nice thing is, in the end, what we will be able to is to also bring this together across the board. Yeah? So what is the influence of maybe changes in the viral genome on the immune response and the host genetics and so on. What I would like to share with you today is a study which is now entitled at the moment as Suppressive myeloid cells are as a hallmark of severe COVID-19. I can tell you that there is more single cell omics ongoing in DeCOI, which I will only tell you that people in Kiel, in Berlin, in Hanover, but also at other places within DeCOI are actually also producing single cell data in Dresden with different foci. So for example, some of them are more interested in the T cell response, others in the B cell response. And this is all done on single cell level. Others, again, longitudinal data. This is all ongoing. We don't have any data, but we have one study where we are basically in the situation that we already can share you things that we also pushed out on the internet. This was an enormous collaboration. I would like to thank these people up front. And very important to say that we used actually two cohorts, one in Berlin and one in Bonn. And I think many thanks go to the people in the clinics, because both were very nicely organized. One is leading there is Leif Alexander from the Charité, but also Jakob Nattermann from the medical clinic in the University Hospital in Bonn. And I have to say, if you want to generate in a very fast time very good single cell data, you need to have a very short and very good collaboration with these clinicians or clinical scientists that do both. Basically, they do science, but they also have their clinics and their patient base. I would also like to thank people that do more the computation, like Emmanuel Saliba, who is also a member of many of the single cell fields, also in Lifetime, Yang Li, who came from the Netherlands, Birgit Sawicki, immunologist, who did some CyTOF work, which I don't show because we're focusing on the single cell work, and Anna Aschenbrenner in Bonn she's really helping to connect the medical, biological, and more technically oriented people, because she really goes in both directions. So having said that, what I would like to show you is basically the setup, what we did. And I think that is also something for me unique so far, to have one study done, but already doing single cell in cohorts, and here even two cohorts. The cohort one was done in Berlin, the second cohort in Bonn, and we even used different technologies. As I told you, there was CyTOF, which I don't show today. We used whole blood, but also peripheral blood mononuclear cells. For those of you not in hematology, immunology, there's different ways of isolating cells. If you take whole blood, of course, you have all red cells and white cells, so immune cells. If you take this PBMC, you only get a certain fraction of the immune cells. That's nice because clinically it's easy to be done, but we're missing quite some cells, and that's why we also decided in the second cohort to do single cell on all white blood cells, which also includes granulocytes. And you will see throughout the talk that sometimes these little changes in the lab make quite a big difference on the information content, which is actually true for many single cell studies, also for other tissues, that this first part here is actually a critical one to be considered once you get these atlases in the end. So we did 10x in Berlin, and we did the BD Rhapsody whole transcriptome in Bonn, and also some multicolor flow cytometry. In the end, we had samples at different time points from different patients with different severity of COVID, a total of 100. Of course, we continue, so we also have already some questions that we have to answer for this study, and we have continued to sample to answer certain things concerning the association of the single cell data with the status of the patients. For COVID, you might all know that there is still a debate of how to call these states. Mild and severe is a very clinical one. Then the WHO said we have 7 or even 8 individual stages. 8 is diseased, 1 is the most mildest one. And I see there's some fluctuation still because we still don't have enough data showing that these clinical categories are really telling us exactly the biology of the disease. So there might be, we might see even some changes there. So with this setting, we just looked into how do we analyze them. So also to integrate that, you have to also make the comparison of the 2 cohorts. So we had a clear plan of what has to be done. There's an overview on both sides. In the cohort in Berlin, they used first Disney plots, for example. There were certain clustering approaches where we decided to do them very similar, but still remember we have 2 different techniques. One is droplet-based, one is array-based, and then bring it all together. And also the data set is richer than we have reported so far, so we left out quite some immune cells that also have changes in this disease. Now, this is work mainly by Bowen Zhang in Yang Li's lab, together with Emmanuel Saliba from Würzburg, where they looked into the first cohort, just in Berlin. You can see on the side it's about close to 50,000 cells, which is a decent size already, or just one cohort. And he did the classical things, dimension reduction, clustering, and then basically with marker genes labeled what we think is there. And in blood, this gets more and more easy these days. But what was surprising is usually what you don't get if you have peripheral blood mononuclear cells, you never get neutrophils, because neutrophils are granulocytes, and by definition, when you have PBMCs, the procedure should actually get rid of granulocytes. But here we had suddenly again some of them. And what you call them, because PBMC is a density gradient, so the lymphocytes, these PBMC have low density, and the granulocytes have high density. So it was very clear that if you see some granulocytes here, they must be low density. And then if you go into the literature, you already know that low density neutrophils can be seen in strong inflammatory reactions. And of course, COVID is one of those. And then very often these cells are actually what we call emergency hematopoiesis. So the body really tries hard to make more and more of these cells, because the bone marrow gets signals to say, like, we need more of these defense cells, and neutrophils can be made in millions and billions of cells per day. So this is a sign that this program between the bone marrow, the blood, and the tissues is on high speed, and that's why you see these cells also in the blood. But what's interesting also in this, yeah.
Sorry for interrupting. This you mentioned 50K cells, right? This was just for the subset of severe COVID patients. But how many of those patients was there? Was there a need for integration? And you also said there were multiple time points. Was this just a single time point?
No, so this is basically the integration of all, and I come to the time points a little bit later. Yeah.
So this is integrated in how many patients?
This is so that these are, I think, was 10 patients in total of several time points. Yeah. And you can also see that one important aspect is, so these cells that you see, this cluster 13 and 9, is mainly coming from the severe ones, which you see on the right side in red. So we don't find anything here with the mild ones. That was already the difference. So inflammatory monocytes are also something that you usually don't see. In homeostasis, you have 3 classes of monocytes: classical ones, non-classical ones, and intermediates. And these inflammatory ones is clearly something that is from disease. And you can again see these inflammatory monocytes in this cohort is mainly from the mild patients. So of course, then we wanted to see what happens in the other cohort. Is this the same, different technology? But again, we see these neutrophils coming up. So this is cluster 18 and 13, similar thing. But here, this was much more rich, and we see these inflammatory monocytes, but we also see some additional monocytes that we're not seeing. And there's also some things I come back at the end. We know that it's not only the technology, but it's also whether you freeze cells or don't freeze cells. Some of the cells have troubles to get through the freezing process. So we have the idea that there's a couple of more that are more fragile, and we just captured them because of different procedure in the cohort 2. Right now, we're trying to figure out how this is if we do the same procedure in both cohorts. So that was nice. Then, of course, you look at genes, and I hope you can see it on the left side. So here's the cohort 1 and cohort 2 for the genes from these clusters that we were interesting. And it was really amazing. If you think about this, we're done in completely different labs under different conditions, different technology. And if you look at the marker genes for those cells, then particularly in these neutrophils, they were almost identical. And that was really striking to us. And I think it's also one of the strengths here to say, like, without many aspects to further look into data integration, the signal in COVID is so strong that we don't have a lot of impact on technical aspects, because the biology is just major here. The same is true for pre-neutrophils and immature neutrophils. For the neutrophils, for the monocytes, we also see populations that are almost identical. And then we realized that there were some more cells that come in the second cohort, and I can explain that to you in a second. So we also looked at these low density neutrophils. If we combine the 2 studies and look at these cells in the different cohorts and also the different patients, you can clearly see that these neutrophils come up in the severe patients in both cohorts. And this is true for the immature as well as the neutrophils. And if you look at these inflammatory monocytes, which are good guys, they only come up in mild, but not so much in the severe. And then we have this other monocyte population that we only see in severe. And this already pointed out to us that you need these kind of single cell technologies to really understand that there is different things ongoing at the same time in cell types that we usually look at as one global population. So having said that, we wanted to, of course, know more about this. So we zoomed first into the monocyte space, and this was mainly done now by Kevin Basler in Bonn, where he looked at this in the second cohort first, because we had more data there. And still the structure is kept, what we know from homeostasis. So we have these so-called CD16 positive monocytes, which are non-classical. We have many CD14 positives, which are usually not the classical ones, but you can already see that with this clustering strategy that there is quite some structure in the data. And then we were, of course, interested, what are these kind of cells? And can be, from the genes that they're expressing, can we actually infer functionality of these cells? And you can. I mean, there's quite some striking findings, which is unique to COVID, not overlapping. Some of the stuff you can also see, for example, in some sepsis patients, but as a combination, as we can see it here, and we have some other evidence as well. This is the combination of things that are happening seem to be somehow unique, particularly to the severe cases. So one thing we see is that, when you take the marker genes on the right side, this FCGR3A, this gene is CD16, you can see clearly that you have these non-classical cells, which are also low in CD14. The other ones have CD14, but then you can already see within the CD14 positives, that you have some of the cells that have loss or very low expression of HLA-DR. And from the literature and from many also functional studies, we know that if monocytes lose MHC class II molecules, they can turn off T-cell responses, which is of course not a good thing. Because then the antigen presentation is more in a context of turning down T cells rather than activating. We have a couple of them that are inflammatory. So you see that in gray in this number 2. These are clearly positive for these molecules, but we know that these cells are mainly seen in the mild patients, but not in the severe ones. And then there's a couple of things that also could be easily linked to the biology of what we see in the severe patients, because many of those have thrombi, they have coagulation problems, they have embolies, and we see a special population in here that has the program to activate platelets, which is also something you only see in severe patients, but not in the mild ones. And this is also a very rare event. So this is, we could say, like, that's the last resort of a immune system to fight a situation where everything goes awkward, to activate things. It's mainly used, for example, in wounds where you have bleeding, so that the bleeding is stopped. You need to really close the vessels, and then you need platelets to be activated to close something. It's a last resort mechanism, but that should not be activated systemically. And here we see it just in the blood, and that's not a good sign. And then we have these cells that are, you could call them suppressive, because I can also assure you they have many markers that are associated with suppressive monocytes. And this is another thing, probably fighting the immune system, fighting the system to shut down, because there is some feedback loops clearly indicating that the system is not going into the right direction anymore. You can also then look at this on a time scale. So for some of them, we had already at that time point some more time points. So this is over time. You see the mild ones on the top and the more severe ones low. And you see also the WHO classification. You can also see that this we monitor that over time, so it changes. Makes sense. If people are going to the ICU, then WHO classification is going up. But you can also see that there's certain subtypes coming up that are not showing in the mild ones. So for example, the mild ones, you have more these rosé ones, which are HLA-DR high. These are the ones that can activate T cells. And this is much lower, and particularly in the early phases, you see in the severe ones, these platelet activating and suppressive blue ones. And this is something we see either very little or not at all in the mild ones. And so there is a clear difference. We, of course, the next steps will be to understand why these patients show up with these changed programs. But it's clearly that the programs that basically switched on in the severe explain why this awkward going immune response actually is the problem of these patients. At that time, it's not the virus anymore. And it's also known now that the viral load is not so much different between the severe and the mild, but it's this wrong-going immune system that's the problem in these severe ones. We were interested in, of course, to see not only the monocytes in the innate immune system, but also the neutrophils, and started with these low-density neutrophils, actually in the PBMC fraction, again, going back into the cohort 1, and this was again done by Emmanuel and Yang and their team. And you can clearly see, if you take only the low-density neutrophils out and subcluster them, that there is the structure. We can really see what is known in the field. So neutrophil experts tell us that there are so-called pre-neutrophils. These are immigrants from the bone marrow into the blood, and usually they should not be in the blood. But if they are, then this means there's a huge output, and the system is completely stressed, basically. Same true for immature cells, but we also saw a couple of the mature neutrophils in there. And then we look for genes that are known for certain functionality, and also known for these genes to be expressed, for example, in sepsis patients. And some of them show up, but not exactly in the same pattern like they are in sepsis, so there's some differences. And we also found some genes like arginase 1, which is coming up in cells that can suppress T cells. Same is true for some of the other factors like FM4 or further up MPO. So PAD-I4 again is one that is associated with platelet activation. So again, these things show up in a situation and only in the severe ones. We don't find them really in the mild ones. Question is, of course, these are very few cells. So what happens with the rest of the cells? So in this case, we went back and in Bonn did some fresh blood cells, so changed the protocol. So now you could even say we used a different tissue because now we're looking at cells that are usually not in PBMC. Here we overlaid that. So we had samples that were frozen, peripheral blood mononuclear cells. Yeah, so this enrichment of these cell subset. Then we did this also fresh, so directly, from the patient, from the floor, directly to the lab, and then processing everything immediately. That is logistically, of course, much more difficult. And the same with the fresh blood. So, Lorenzo and Elena got the call from the ICU to take the blood and then process it immediately in this usually very long days because you're not getting the blood the time when you would like to have them. And what's very clear is that the granulocytes only appear in this ones, really. Yeah? This is the major part here, this 46,000 granulocytes in the middle in red. You can clearly see this big blob here, and you see these two smaller ones, and probably you can guess already these smaller ones are the premature ones that we saw already as low density in the PBMC fraction. You can also see them here a little bit in the middle, in this dark blue one. While you can see clearly that the majority of the granulocytes are lost if you do the PBMC, which is normal because that's what the procedure is supposed to do. So when you look at this now, we subclustered out basically now only the granulocytes. It's a rich space, yeah. So you can see again that there is much more substructure than it has been thought. For the non-immunologist, neutrophils in the textbook are these dumb cells that live a day and then die. And kind of a suicide job if something urgent happens somewhere, you get a wound or you get a local infection, an abscess or so, they go there and then they die and release all their tools. It's not true. Neutrophils are much more diversified, and this only comes out now actually in the time of single-cell RNA sequencing. So we looked, of course, again, this was the integration of all patients, but then we asked, which clusters comes from which patients, and there was a clear picture. You see in the middle, there is a cluster that comes from mild. There's a large number that we only see in controls, which were, people from the same clinics, but with other diseases. Then you have two major clusters that only come from the severe ones, and then hard to see, but this little bow there, these two populations, which again are the premature pre-neutrophils and the immature neutrophils, they come basically only from the severe ones and the late ones. And we then basically
to understand. Sure. I think it's pretty exciting because that's stuff you don't see in PBMCs, right? And I guess in HCA, immune cell atlas, and so on, I think they also don't have granulocytes included.
No. People always thought, it's not necessary because these cells should all be the same, because if they live only a day, why should there be such a big difference? And we just learned that now, that for many diseases, actually, these cells are probably more important than we think. It's a little bit
exciting. Is that something you cannot get with droplet-based assays? So was this only from your Rhapsody, or did you also do this 10x from Berlin?
We have not done that in Berlin because of that. So I'm pretty sure it's not completely lost. So there are some improvements now with the 10x. So I think that's fine, that you can grasp also some human ones. So in comparison to what we have shown before, but I'm not sure whether we get all of them. So I would still say the gold standard for granulocytes is a microarray based technique, because these cells are fragile, and even under these conditions, they're really hard to handle. So I would say, like, the more gentle you can be, the better. So for the moment being, I would say people who would like to do single cell on granulocytes should stick with a plate-based assay. Then they're for sure having a good representation of what is in the sample.
I mean, I'm sure there's going to be later discussion on basically those variances that you see in control mild severe, so I won't ask for that, but I think it's really exciting that you look at the subpopulation.
Yeah. Yeah, there is, but that's also because we have, by purpose, we didn't use controls healthy people, but we wanted to have some other diseases. So these are also sick people from the hospital. Yet, you know, in this compartment, it seems that they're very different from COVID, and I think that's the important message here. You can have a, you know, an exacerbated disease, a chronic disease with inflammation, but this is very different to what we see in acute COVID. Now, you can then basically look up, you know, which clusters are basically at which time and from which patients. That's what we here just summarized on the right side in the heat map. And then, of course, it's again interesting, you know, what kind of genes are actually expressed, what is the functionality. And again, I would like to point out that in the severe ones, we find things like PD-L1, which is known to be on suppressive cells in the innate immune system. You can call them different types. There's a whole field on myeloid-derived suppressor cells. PD-L1 is a major molecule in these cells. Everybody knows in the field that I'm not such a big fan of this word myeloid-derived suppressor cells. I think we don't have to stick to namings. We could just say, like, we know that cells with this molecule are having suppressive functionality. The same is true also for some other things like CD64. So we have an enrichment of molecules that are associated with effects on other immune cells that we would call overall as suppressive in the severe patients. And of course, that's not a good sign. Yeah, so these patients have a problem to basically support an antiviral immune response. What we also can see in the lower part, it's a little bit difficult to say. So with these red boxes, this was work done by Nico and Jonas, where they looked carefully of certain genes. You know, in the immune system, sometimes single genes make a big difference. So PD-L1 is already some, you know, either you have it or not, and you have the opposite functionality. But there's a couple of more of those which are more recently described, like, for example, this ZZ3H128 gene. There's not a lot known about it, but it's also associated with changing functionality of immune cells quite dramatically. And you can see that this cluster 5 doesn't have that, yeah, while cluster 2 and 0 has this gene, yeah. And it's surprising, you know, that we have cluster 5 is the one that we see in the mild ones, but the mild ones have everything you need for interferon response, which is more on the left side, like the IFEs and Ly6E and ISG15, so all these big blobs there. But the 3 that are missing, yeah, these are the ones that would be inhibitory. So it's very interesting that in the severe ones, you're switching on interferon response genes, but at the same time, a program that is more inhibitory. What was also striking for us is that in cluster 9 and 8, which are these pre-neutrophils and immatures, you have other mechanisms that are inhibitory. So there are at least 3 populations of cells, and then even 2 and 0 is like early and late, where you have different mechanisms of regulation or suppression of the immune system in different compartments of the cells. Yeah? And again, I would say if you once have that information, you can probably also identify these things in bulk, but it's of course much easier. You have the resolution first and then interpret your bulk data with that knowledge. Yeah. So we're coming up basically with the neutrophils in COVID and have like a nomenclature for those as a reference for the moment being. And of course, the next steps will be to integrate more and additional data, because we're just striving at the upper level. We know that, but I think it's a very, very important starting point to clearly state that we should not leave these cells out. Clinicians would tell you immediately, because, for example, in the clinic, the ratio between neutrophils and lymphocytes is already used to identify patients that go into a mild or versus a severe situation, and we can reflect that now with much more biology on this level. What we also did in the end is that we looked to identify what is changing. Yeah, so again, we used here as an example, and of course this can be done now with all the cell types. So we use these 3 clusters. This number 5, which is in mild patients, the cluster 2, which is in severe, and late is the cluster 0. And it's really striking if you look at the early time points. Yeah, there's not so much difference between the mild and the severe, but if you look carefully, it's a very few things that change. And I think we have an Achilles heel here of identifying the severe program to really target that then also therapeutically. So there are some biological mechanisms that we're still working on right now that we think makes the difference. You know, what's not different is both of them make a very strong interferon response, which you see with all these IRFs and stats and so on. But there are very gentle differences, and these are the things that we need to address. What we were also surprised is that if you look for later cells, so this cluster 0, we're basically losing biology, and that's also an interesting fact. So these cells stick around longer, and that's actually we have seen now a couple of reports has been seen in some other diseases that these cells actually increase in lifespan, but then they stick around and are somehow misguided. Yeah, so it's kind of you don't want to have these cells around because they block productive immune responses, but they're sticking around because they could upregulate their machinery to survive longer, and that's also a very bad thing that happens only in other diseases towards the end of life. So a lot of signs that you know, we think many of the things that we see in the clinics, these granulocytes and these monocytes will play a central role, and I think this is a good target cell for going for these severe cases. So I would like to bring this to an end and say, like, so far we have 210,000 single cells in peripheral blood of different flavors, so whole blood, PBMC. We probably will have for a revised version quite some more cells because we have sampled more. I think the very strong thing here is the dual center study, and I'm very glad we could do this, and DeCOI made this happen because of the very open way of sharing information, sharing protocols, but also understanding that during this crisis it's very important to work together, and it makes it much more stronger. And honestly, it was so much fun. It was an intense time. It was very much fun with Leif's team and Birgit's team and Emanuel's team and Jakob's team and ours to really work on a daily basis and meet in meetings, but really saw the data coming together. That's something, with all the crisis, but there's also some good things, and that's what happened here. We have the biology, so we have certainly a good immune response that is inflammatory monocytes, very high in antigen presentation in the mild patients, but then you have this multiple mechanisms and multiple cell types and cell states that come up that basically turn down everything into the wrong direction. And I don't name them all here, but this is not good, and in this magnitude has not really been seen in other diseases yet. And with this, I really would like to thank again the team. I don't have pictures, there's so many now. We're still collecting pictures to have a nice one. It was very much virtual because at different places. But again, I would like to thank the team in Hanover, the team in Berlin, the team in Würzburg, and in Bonn and beyond. We had exchange within the DeCOI, and I think I hope a little bit of that spirit that to share things very early on and do things together will keep even after the crisis and once we're a little bit more normal again. And I thank you for listening.
Thanks, Joachim, for a nice presentation and for a great overview of a very important and timely data set. I think very excited to many of us. We have time for questions. Please ask, and then I think I can turn up your microphone. There's a first question from Wolfgang. I hope. I have to click here. Wolfgang, can you?
Do you hear me?
Yes.
Hi, great. Thanks. That was wonderful. Pretty cool. Technically minded, I would be really interested in how the 10x versus the Rhapsody thing kind of turned out I mean, obviously you have a big biology, as you said, right? So that both shared the signal, but on a more fine-grained level, because I keep getting these questions asked, should I get a Rhapsody, and I've never had an answer because I don't know.
No, it's very clear both technologies are advanced now. Also, I think where the Rhapsody made a big step forward is when they introduced the whole transcriptome. They're very reliable. Yeah, that's important, particularly when you do this clinical work. You don't want to fiddle around. I mean, we have another nice technology set up in the lab, which is Segwell, but it's handmade, and we decided not to do it because we were not sure whether we should go for something where we know it's cheaper for us, but if something goes wrong, then it's not that standardized. Both techniques are concerning that, fine. The very fine-grained technical comparison, I mean, as you have seen, we dealt with that really as two separate studies by purpose. You know, we wanted to understand whether if you do a dual cohort study, you would come up without integration to the same results, because you could have, you know, technical impact that lead to different results, and you could heal that by integrating them. I think the integration is something that we want to do later to see whether we get even better or what that means. And that is something that we learned from Malte Lücken in Fabian Theis's lab. That is a data set that is fun to study that now. The direct technical comparison is still ongoing. Yeah? It's not part of the initial report, but it's actually something we should do and find out, because this is we can benchmark that. We still would like to do a couple of samples where we do both in one sample, not only two cohorts. Yeah? And that's planned because we have a 10x also in Bonn, but it will be not part of this one. So I cannot give you the final answer, but just seeing how, you know, you remember this one where we looked at the neutrophil genes. I mean, first place when I saw the picture, I thought like, hey, you have them twice the same. And then Jonas told me, no, this left is Berlin and right is Bonn, and these are, you know, coming from two different techniques. That was mind-boggling. I mean, it's really, really cool. So technical things will come later.
Cool. Yeah, that helps. Great, thanks.
There's another question from Malte, who I will now bring in here.
Hi, thanks for the really, really interesting talk, Joachim. Unsurprisingly, maybe I also have a question about integration. But here you presented quite a lot of things from like multiple patients, right? So you mentioned 10 patients in the one study. Were you required to do any removal of the batch effect between these individuals, or because you found really nice kind of populations and they do separate out disease and healthy, and you do find even like markers for them despite this complete separation. So I can believe all those populations, right, that you find. Would it be interesting to see like if there might be an effect from batches?
Yeah, absolutely. I mean, and Malte, you're the expert in that, and we should make this data set very quickly available to you so you can study that in more detail even. I can give you the first glimpse, you know, so we have more correction was necessary for the 10x than the Rhapsody. Yeah, and if I'm not completely wrong, because we worked with that, but we did not correct. We just used the data as they came out of the machine for the Rhapsody. And we see that over and over with micro-well-based single-cell technologies, that you, between the different arrays, and you just have much less batch effect. Yeah, so here we did not even use any correction. Yeah, what we did is we did multiplexing. So even more workload on the lab people. We had always AB antibody-based multiplexing of all these samples, and of course that makes life easier. But even samples from one patient done on two arrays was not corrected because there was nothing to be corrected. And like this, I think you can integrate patients quite easily. Yeah, but you also know, Malte, we took, you know, we spent many months, you know, to make sure that we're going this direction, because we know that we make your life as a computational person much easier if we take care of all this as much as possible, because you can have more fun on doing the biology rather than only on correcting things if we in the lab do the better job, right?
That's really impressive, how you keep managing with the sequel and now with the Rhapsody as well to get very, very small batch effects. Maybe as a quick follow-up, could it be that you're sequencing very little, or are you still sequencing quite deeply?
Not to spend too much time, we sequence much to, you know, we sequence deep. We can even go deeper, but you know, we discussed in the group, there will be not much more information coming, but we spend quite some sequencing costs, not just to spend time.
Okay, there a few questions also in sort of the texting. People have been congratulating you for a nice talk and so on. There was one question, congrats for that as well, obviously. And there have been one question, if at some point you make a preprint available for that or potentially data.
Yeah, so if you go in medRxiv, there's the preprint at the moment. Yeah, and right now we're working very strongly on questions that we have been asked by reviewers.
I checked on bioRxiv. It's in medRxiv. Okay, cool.
Yeah, no, it's med. You know, we decided to go in medRxiv, but it's there. But if somebody doesn't find it, you should find it. I mean, you see all the names. It should be easy to be found. And if you have questions, you know, there's the email. Just send me an email, and we're trying to answer more.
This next question from Carsten Ma.
Thanks, Joachim. Fantastic talk. I have two questions. The one is, how unique is that profile you showed us for COVID-19 as compared to other viral or bacterial infections? Mm-hmm. And the other one is, you showed these subclasses of neutrophils. How sure can you be that these are subclasses that emerge in every patient and are not an effect, you know, mm-hmm. of merging the patients together?
Mm-hmm. So that's a very good point. And here I can just tell you that we have more information from other patients with other technologies. That's why I'm very sure. Yeah, so we have done a large German, but now even European effort to also get samples from larger studies where we can do not so fine-grained single cell, but we did PAX gene. And of course, if you have both informations, you can actually enrich PAX gene a lot. By the way, you know, we had two teams in the lab. One was working on single cell, the other one was more working on the PAX gene samples. And I have to confess, they also got a lot of biology just out of PAX gene. I'm very proud of them. So there we have many more patients, yeah, and so I can just tell you that in the severe patients we find molecular phenotypes, yeah, so that there are differences between patients, but then we find patterns over and over again, yeah, and so I think I'll completely trust here, and we're now also adding more patients to the single cell, that we also see these molecular phenotypes that are distinguished even in the severe distinguished patients. And of course, that's important for therapy development, because what we think is that the drugs that are out there might be better targeted if we know which molecular immune phenotype you have. Not all of the drugs that are out there, you know, to what we did for drug prediction on these cells would basically benefit all patients would benefit. There will be a subgroup, but not all of them. By the way, we predicted dexamethasone 4 or 5 weeks ago, yeah, and it's really pushing down these cells that we find not to be good in severe. Yeah, so we were very pleased to see that if you do clinical studies that this comes out, and we have many more drug targets predicted now, so that would be another avenue. What we have to discuss, and I'm happy to, you know, if people have ideas. Right now we did the drug targeting on bulk, but maybe that could also be done on single cell.
There's one last question from Maren, and then maybe we should wrap. Wait, I'll have to. Here, Maren, here you go. Maren Büttner.
Thank you for the excellent talk. I was wondering about the disease progression. So when you say you have patients with mild and severe outcome, I was wondering if you can actually already see in a mild patient, well, how to say, the disease progression is way further in severe patients than in mild. So I was wondering if you have a patient that has still mild symptoms because before he goes really bad, that the immune system looks different there compared to someone who is mild all along.
Yeah, wonderful question. I think we will learn more from other groups as well. I also know that the Kiel group is very much focusing on, and they are closer to that. I can tell you that we have also 2 patients where, you know, once we knew that this is important, and then even these more suppressive cells show up earlier in the severe ones, we wanted to get, because we wanted to get earlier time points, which of course is difficult in the severe ones, yeah, because very often they come late and then you don't know how long was the period before. But I think now we have 2 patients followed from very early onset. So they came very early and then turned out to become severe patients. And we have followed them with single cell over 10 or 15, between 10 and 15 time points. Yeah. So, and we hope that we can answer some of that question. Yeah, because we have early mild, and then we have these ones now over time, and then hopefully find those genes that tell us why these guys went into severe. And then you can actually take these genes and validate in larger cohorts again.
Beautiful, thank you.
Thank you for the nice presentation again. A lot of cool large scale data, different technologies. I think a lot of fun be potentially ahead on that. I'm looking forward to hearing this is on medRxiv, so I'm sure people will be picking it up, and I think maybe we can tweet afterwards around what the link is, so that people see this as well. I think there's some nice method aspects that could still, of course, be addressed. And you mentioned data integration, and I think that would be very, very nice. Maybe projection onto HCA, immune cell atlas, different type of things. And then there's also this differential testing, because I know that particularly, like you're like a immunologist, often coming from looking at FACS-based data and looking at relative populations, how to actually deal with these population-based effects, in particular over time, as you saw. I think that's not yet fully addressed on the method side.
No. That's what I said. We had already, when we pushed out the medRxiv, some people that said, like, can we have the data? And I said yes, but let us basically, we have been asked to put on a couple of specific additional data first, some more patients for certain time points, for example. We would like to finish that so that we have one complete block, and then make it available to people that are interested to study these aspects. But right now we have an interim phase, and I think very soon we have it finished, and then it's the time that we can have a situation where we can start sharing for those that can further develop things and so on.
In any case, I think it's a fantastic start for this DeCOI consortium, and thanks again for a nice presentation, and also for doing the organization for this whole thing in the background. With this, thank you everyone for listening to our bi-weekly SCOG webinar series. 2 weeks, Julio will talk, and I hope to see you all there, and thanks for the nice discussion. See you, everyone.
Thank you very much. Thank you, everybody, for listening.